I know. I'll remember half of it. You and your farm? Someone, at least 6 people. Good morning, folks. Welcome. Thanks for coming on another torrid day. I'm Dan Sarowitz at the Arizona State University Consortium for Science Policy and Outcomes. This morning, I am truly thrilled to have Lisa Marginelli here to talk about her incredibly interesting, cool, wonderful new book, UNDERBUG, and to have her to throw her into the clutches of an interlocutor, Richard Harris, who will, together with Lisa, have a conversation about what her book's about, what it might mean, and hopefully, before they're done, draw you into the conversation as well. For those of you who have come to these sorts of events before, I don't believe in big, long introductions, so I'm going to stop and turn it over to Richard, who will get us started. We'll do the standard sort of thing with you guys will talk for half an hour or so, and then afford opportunities for you to participate in the conversation. When we're done, after about an hour, there are books in the back. Lisa's happy to sign. You should be happy to buy. And then we can all go away happy to our torrid lives after what I hope and I'm sure will be a really fun interlude and great way to start the day. So thanks for coming. And Richard, take it away.
Thanks very much. You guys can hear me, I hope. Thanks. Excellent. Thanks all for coming. I am delighted to be here. Dan asked me to read the book and participate in this, and I must say it was a great treat for me to do that. And I want to start by just asking you a little bit about how you found termites. I mean, you were doing energy and reporting, right? And
right.
Termites seem to be a pretty sharp left turn.
Yeah. Well, I think the termites kind of found me. Is this sound okay for you all? Okay. The termites kind of found me. I was doing energy reporting and working with the New America Foundation doing energy. And I heard that there was a project out at I was living in California. I heard there was a project at the Joint Genome Institute, which is a Department of Energy lab in Walnut Creek, and that they were sequencing the microbes in the guts of termites to try to find clues for making biofuels. So it started as this very idealistic plan to replicate the termite in a steel tank. And 10 years later, that sounds ridiculous. But 10 years ago, that sounded like a reasonable idea. And so that actually sort of frames what happened in the book. Is I got into termites, and then the more I got into termites, the more I got into termites. But also this question of how do we take their stuff and use it in some way that we can use also became an obsession. And also, how is our framework of looking at a termite as as a solution to our problems, when for most people a termite is their problem, So that sort of became the obsession of the book.
Yeah, and why? Termites are curious creatures. Why would Energy Lab want to go after the termite gut as a way of producing energy?
Well, termites can eat wood and they can eat grass, and the cellulose and hemicellulose in wood and grass is full of sugar. So we should be able to use it, but it's locked in kind of a chemical cage. And inside the termite guts, the microbes have the ability to, they have enzymes and sort of Swiss Army knives that they can wreck the structures of the cellulose and pull those sugars off and then ferment them into some kind of fuel.
Yeah, and I think that's one of the interesting themes in the book is to sort of look at, it's not actually the termites doing this, it's
their guts.
Critters in their guts. And not only these bacteria, but what I was fascinated by was some of these weird other organisms in their guts that aren't even bacteria. I mean,
they're protists.
Protists. So let's see, I wrote down a couple of names, ones, Trichonympha and Mixotricha paradoxa.
Yes.
So just, I mean, you could, I mean, obviously there's just this weird world going on. Before you even sort of start to layer on, you know, who's in charge? Is it the termites? Is it in the guts? I want to get to that in a moment. But tell a little bit about these weird protists that are hanging out in there.
So there's two types of termites. There's higher termites and there's lower termites. And the so-called lower termites have protists in their guts. The protists are, they're not an animal and they're not a vegetable. They are, some fungus, I think, are protists. But the weird thing about the protists in the termite guts is that, so they're a little microbe, you know, let's just imagine they're like this, but once they get into the termite gut and they live there symbiotically for 100 million years, they change in very weird ways. So the protists in the termite guts grow to be like 1,000 times their original size. They make a structure that's repeating and they sort of jam it onto themselves so that they're much larger. That's sort of like bolting a lot of IKEA bookshelves together. They take their initial genome and then they just expand it. But then they also have partnerships with sometimes 5 other genomes, 5 other creatures, sort of integrate themselves into the protist. So Mixotricha paradoxa, which means the crazy paradoxical creature with the mixed up hairs, actually has 5 genomes cooperating as one. It has a sort of an extra energy cell. It has a bunch of hairs. What look like the crazy mixed up hairs are actually separate creatures who have integrated themselves into the protist. And then they also have a lot of little creatures around their mouth.
Yeah, and I bring this up not only because it's weird and wonderful, but because I mean, one of the central questions about your book is sort of who's in charge. Mm-hmm. Are these protists in charge? Are the bacteria inside them really calling the shots, or is it the termites that are sort of farming these things inside, mm-hmm. or is the mound in some ways that they build in charge?
The creatures that build a mound are higher termites, and they have, in addition to a bunch of bacteria in their guts, they have a fungus that digests the cellulose for them. And the fungus is in the base of the mound usually, and then there's the mound up above. And the termites get grass for the fungus, which can be like 9 times the size of the termites in the mound. So the fungus has heat, it has gases, it may have chemical instructions that we, being humans, are too dumb to smell, but they might mean a lot to the termites. And the termites collect grass from a large radius, bring it back to the mound, shape it into this wonderful, weird organic shapes that the fungus likes, and then they inoculate each ball of grass with fungus. And then the thing starts to sort of sprout fungus, which they tend, and they suck the sugars off and run it off into the rest of the mound and distribute it to their friends, the other termites. That's pretty weird. So who's in charge there? Is it the termite? We assume, because termites are more sophisticated, that the termite would be in charge of the fungus, but it's possible the fungus is in charge of the termite. And then when you talk to...
the fungus is telling the termite...
Possible, yeah. And possibly the things inside the termite's guts are telling the termite what to eat. Just as maybe you're drinking coffee because your gut microbes are telling you that they would like some coffee. Or maybe that's why you crave salt. And we're increasingly aware that our own gut microbes have some sort of connection to us that might be uncomfortable.
And we've started this conversation much the way that your book starts, which, although the title says An Obsessive Tale of Termites and Technology, you really start off talking about termites for a bit first before you sort of turn the corner and say, so what does this have to do with technology? So what does this have to do with technology?
To back up slightly, I mean, if you're talking about who's in charge, what does that have to do with technology? Well, it's really interesting because the two things that people are trying, there's basically three things that people are trying to get from termites. One is biofuels and recipes for biofuels, and the other one is algorithms for swarming and building. How would you build autonomous robots that can build the way termites build a mound? So when termites build a mound, they can go 17 feet up. So that's a termite this size. That's basically the Burj Khalifa. And then, but they do it without any architectural plan. So how are they doing that? They must have internal rules that say, I'm a termite, I grab a ball of dirt, I run around with it, I follow an air gradient to where it's fresh, I drop the ball of dirt where there's a smell from other termites, and that makes a stack. So we can get as far as thinking through to this stack. We can't get to the rest of the shape, really.
But it sounds like a simple algorithm.
And we have built them in labs. The scientists from the BOWC Institute who are in the book have built them in labs. But we kind of need to go to the next level to figure out how to build a real structure. So that's the second thing. But also, when you look at swarming, who's in charge of swarming? It's not, there isn't a mastermind. There's all these mini minds. And what happens? You know, fish have when fish, school, yeah, right, I was thinking flock. But when fish school, sometimes an eccentric fish will have an idea of going off to the side, and the fish have sort of correction systems so that they can figure out who to follow. So these systems have corrections and feedback inside them, but we don't know what they are. So as we build swarming robots, we need to kind of be sure who's in charge. And hopefully also for weaponry, more than hopefully, in a very sincere sense, hopefully humans need to stay in the loop of deciding when swarming weaponry works. So that's one of the implications for technology. And then the third thing that people want from termites is understanding the fertility of the soil.
Yeah, well, and I want to get back to the schooling thing for a minute, because one of the revelations for me in this book was that we've always heard about the hive mind, where it's no one's in charge and everyone's in charge and so on. It's more nuanced than that, right? It's not as though everyone is doing exactly the same thing. There are lazy termites, and there are termites who are, for no apparent reason, leaders, right? Right.
They're charismatic termites. This is a really weird idea, partly because our idea of termites, early Western explorers who looked at termites saw them as a model of society that sort of showed that, you know, kings should be in charge. And then our model of them in the 20th century was really that they were factory workers. So everybody was doing the exact same thing all the time, and they were all being productive. But when you actually track the termites, they're screwing around. Like only 20% of them are working, and out of a little petri dish of them, 2 or 3 are charismatic leaders, and they're getting the other ones to bring dirt, to dig up dirt balls here and put them down here, and then some are just sort of standing, doing absolutely nothing, and some are just running around. And some are screwing around. They're just like running back and forth between the other ones. And so what appears to be order, because we have an idea that this must be orderly, is actually not. It's like chaotic. So what's going on there?
And the engineers who thought, oh, we can build a mechanical termite based on the simple algorithm of stacking dirt or whatever. Turns out they are not including this rather bizarre behavior into it.
Right. But that made the roboticists think, like, well, why did I think that? This is really interesting because it was an insight into how the roboticists think. The roboticist Kirsten Peterson in the book, who invented the tracking system for the termites, then said, I build robots all the time. I know robots aren't identical. They all have cheap sensors, and some are stronger in one thing or broken in another. And so then she got interested in building societies of termites with broken sensors, where some would become smart and others would be dumb but wouldn't know it, as she put it. Yeah.
So tell a little bit about this effort to build these robots. How far along did they get? How good were these robot termites at actually doing termite things?
Well, they say that they're at the very, very beginning. On the other hand, they have a group of 3 termites. They're not termites, 3 robots that they call Termites robots. They're square. They don't build with dirt dollops. They build with specially built blocks that they carry on their backs and thwack into place. And
with the help of magnets, to
yes, magnets and all
so there's
kinds
a lot
of
of cheats.
Yeah, there's a lot of cheats, and yet they can actually. The three of them can build different shapes, depending upon the sort of individual instructions that they're given, and that's very hopeful. And they can build for about 8 hours before they kind of things get before their batteries run down and everything is kind of over. So in a way, we've never had anything that could build that way. So this is a big deal. On the other hand, they're not going to be taking construction jobs anytime soon. And they're not building with anything mushy in anything. Their environment has to be so finely tuned that they really can't work outside that environment. When they leave the building or the corner of the building that they were made for, they no longer function. Just a different room tone starts to screw up their signals, a lack of black tape on the floor. Someone jumped into an arena when they were taken out and broke one of them. And you need not just a PhD, but a postdoc to fix them. So they're not going to be showing up on a building anytime soon. And they can't build in disasters because they can't build with a mess. And building with a mess is really what we want to, if we're going to do this.
Yeah. But these guys actually went to Namibia, right?
Yes, many times.
To try to understand exactly how real termites did their jobs. What was that like? Because you were there with them for some
I was.
at least some of their adventures.
They were kind of amazing because I had been hanging out with biologists who had spent years observing the termites, and they didn't have, they weren't satisfied with their level of insight, but they had a lot of thoughts about what they were seeing. When the roboticists arrived, they saw the termites as a black box, and one of them they had designed this laser scanner, and he was going to scan the termites building on dirt, and then he was going to digitally remove the termites, and he was just going to analyze the dirt balls from point to point in the laser scanner. So he was actually thinking about the termites as not there, which was really interesting. And they thought of the termites as being potentially modeled by robots, and which is an idea going back to Descartes, But once they realized back in the U.S. Years later that they actually were individuals, they had to rethink, and that termites also have memories. They had to rethink how they were dealing with them.
Do you think the roboticists were humbled? I mean, did they came and think, there's a black box, all we have to do is look at inputs and outputs, and we can just model that, and it doesn't really matter how the termites work. If it's a simple enough algorithm, we can just toss it into our machines and go to town.
Yeah, I mean, they weren't as simplistic as that. They were actually very complex people who spend a lot of time studying complexity, but they had this hope that, in retrospect appeared naive. I have to say that the leader of the group, Radhika Nagpal, actually had already encountered weird things in biology in the lab, and she was aware that it wasn't going to be simple. Her hope was to abstract and abstract and abstract, and therefore arrive at the algorithms. But it's sort of like everybody has to learn how weird biology is for themselves.
Yeah, and well, that story was playing out in the Wyss Institute in Harvard area. You were also, there was a sort of a parallel story taking place in the Bay Area, in Berkeley. And they were after another element of termites. Tell us about that.
So also in Berkeley, there was something called the Joint Bioenergy Institute, which is run by Jay Kiesling, a synthetic biologist who'd had a lot of success making a synthetic version of a malaria drug called artemisinin. And they were...
And synthetic biology, we should explain, is basically engineering...
applying engineering techniques to biology in the hopes that you can sort of boot up a cell with software, which is genetic code.
Yeah. So
more complicated than that, but
but the idea is it's a step beyond genetic engineering.
Yes.
So it's very deliberate engineering. And why were they interested in termites? I guess we talked about.
Well, they really wanted to build biofuels. So this was a government-sponsored bioenergy institute, and they felt that we can take information from termites, we can take information from compost, we can take information from shipworms who also eat wood and they have microbes that eat wood, and we'll take them together. And we'll get, just 200 or 300 of the best scientists. We'll divide up the process into specific, we'll treat it like a physics or an engineering problem instead of a biology problem. We'll have different teams working on different aspects of the process. And after 5 or 10 years, we'll have a biofuel that can compete with gasoline.
Grassahol.
Yeah, gasoline. Yeah,
Grassoline, yeah.
Yeah.
I've heard both terms.
Yes.
But yeah.
Yeah.
And they were thwarted also by the termites.
Yeah, they were thwarted by the termites. The termites are much too complex. The termites got booted from the problem pretty early on, although they continued to sort of refer to a table of the metabolic things that happened in the termite gut that's like, it had like 1,000 different reactions, you know. It's like this diagram that looked like a subway map from Tokyo, basically. And so they, but the goal of building like an E. coli or a yeast cell that could dissolve cellulose and make it into grassoline was just thwarted again and again and again. It turns out the cells have internal controls, and they were able to when they put the right code into the cell would say, well, I don't feel like putting out that much of that stuff, so I'm going to divert my carbons over here, and that's that. And so these reactions would get shut down. And that took a long time to figure out what other signaling there was in the cell. They didn't know if the cell had a memory. I mean, whoever thought of an E. coli having a memory? In our view of them, they don't have memories. But
but this gets back to what we were talking about at the top of the conversation also, which is that inside the termite gut is not trivial, right?
No.
These bizarre protists and this collection of microbes and so on is not just a lot of it sort of useless. It's a very complex system, right? And they decided ultimately maybe chemistry is easier than biology and trying to get at some of these things.
Yeah, some of it. Actually, they were successful. They created 2 biofuels, pinene and limonene, but they're like $20 a gallon, and they are useful to the military for missile fuel.
But again, not based on the termite gut.
No, not based on the termite gut. The other thing that happened to the biologist was that you can read their papers, and they start having these sort of existential sadness in the papers, like maybe we can never, maybe we can't abstract biology that way in a way that our brains can understand. These sort of sad things about are we hampered by our brain's ability to understand this so that we can replicate it? And I'd like to ask that the biologists who I was working with whether they had written that sentence.
Yeah. And before we get to questions, I wanted to touch on a theme that you weave in throughout, which is sort of what this means for us as human beings and humanity and so on. Mm-hmm. And maybe if you want to open to page 222, there's a little passage to that, just to give because there's some really lovely writing in this book, and I just wanted to grab a really short passage that grabs that. But you need to sort of set this up.
Which one? 222 on swarming drones?
Yeah. First of all, set up the swarming drones for us, because this is one of the consequences potentially, is like getting this mob behavior to actually be militarized, right?
Right. So a lot of military strategists have been very excited by the idea of swarming drones, because you could potentially have 1 billion of them that cost $1 a piece. And then with that, you could sort of flood areas, and they could be used for assassinations or immobilizing people, or for finding information, or for search and rescue. But this concept that they could be just $1 a piece is very, very tantalizing. And you see in the white papers, you see again and again, sometimes very outdated ideas of what swarming insects do, used as proof that we can do this in the battlefield. And so it's kind of weird to read the papers and actually know, to read the white papers and know kind of what the science is. We are doing the same thing that we did back in the 1700s, where we said this is a perfect society would be one with a king and a queen and soldiers who have to be fed by the workers, and this is what a society looks like. And we're doing the same thing, taking ideas from insects, putting them in our white papers and saying this would be a perfect war.
So if you, in terms of reading, if you start at 221 and just read over to the top of the next page.
The BOWC Institute also was working on a robo-bee, which was a very small bee that was folded in an origami kind of way. It could fly kind of. They were making them so that they could swarm. It's a wonderful project for a scientist to work on. The programming language was called Karma. And the scientists really had to keep themselves from thinking about how these things might be used. So the popularity of the robo-bee as a military concept demonstrates that one of the oldest tactics of war, dehumanizing the enemy, is being abandoned. In Namibia, back in 1904, the German General von Trotha used, quote, extermination and, quote, cleansing patrols to suggest that the victims of the genocide were not people, but vermin or insects. But the swarming drone turns that, and that's something that's talked about earlier in the book, the swarming drone turns that old paradigm on its head. We are now presenting ourselves to our enemies as technological insects and dehumanizing ourselves. The intent may be to show total technological domination, but we will also demonstrate that we do not value our shared humanity. Should I keep on going?
No, I think that was a key line. But I mean, because that sort of you sort of weave in and out of, sort of stepping back. You're not only trying to understand sort of orders of magnitude in the mounds and so on, and the technology, but sort of what this means for us in our humanity.
Yeah.
I think that's a key moment in the book. But there are others like that sort of these things come up. You're stuck in a traffic jam at one point, and you're thinking is Google Maps or Waze or whatever.
Yes. I am the robot. I mean, I've been watching swarming robots all day at the BOWC Institute. I left. It was raining. I didn't take my usual way out of Boston to go north, and I ended up going to my phone, and I got directed to a traffic jam on Route 1, and I was sitting there, and suddenly I was the swarming robot, and we each were following our own algorithm, which was to leave, and could we someday be coordinated for the good of everyone to go rushing out of the city? Like, that's the big dream. We would love that. But at the same time, what does all of that mean? Where does that leave us as humans, or what do we need to, how do we, one of the big things I thought was, like, in this business of trying to become termites technologically, we actually need to think about what it means to be human and how to be more human in dealing with each other and solving problems that are human problems rather than simply throwing technology at them. So the lesson of the bug was to be more human.
This sort of thing. Let's take some questions, and I have more, but I wanted to open up the floor. And while the mics,
can I have moderator's prerogative to ask the first question?
You can, but before you do, let me just make a comment that I would like people to ask succinct questions, not to opine endlessly, and I will actually enforce this with a little bit of an iron hand. But
can I have moderator's prerogative to not ask a succinct question?
All right.
No,
let's see how you do.
I will behave.
Yes. But, at any rate, yes.
I mean, the book raises so many really interesting, big questions. Looking at this little thing, can you just talk a little bit about how it changed the way you think about science, what science is, and kind of what it actually means to learn to know things scientifically?
So, and then I need to leave time for questions.
Yeah.
Okay. So basically, I think I went into this. I had been doing reporting for years. I had reported on energy. I had reported on scientists. I tended to take the idea that scientists were kind of in a special world, a world that an idea that the scientists themselves subscribe to. They follow special rules. They have allegiance to their labs. They have allegiance to their field. They self-police. And and I thought that one scientist was the equivalent of another. So if you said, I thought a synthetic biologist and a roboticist and a biologist who's a physiologist might all be on the same continuum. In fact, they're all looking for very different kinds of data. Their idea of what data is very different. Their idea of what success is very different. For the roboticists, data is repeatable trials that become predictive. For a physiologist, it is to watch something and have an understanding of how evolution might have affected the ultimate structure that they've built. So these are very, very different ways of analyzing the world. And once you throw the synthetic biologists in who are actually having a struggle over predictability, which is the topic of Richard's book, Rigor Mortis. There is a struggle going on within synthetic biology with the old biologists, or biologists who are quite young, actually have been trained to work at the bench as though they're like in a guild. And there are physicists coming in and saying, look, we've standardized this stuff in our field, and we make people follow protocols, and we don't value individual cooking at the bench at the workshop. So there's all this stuff going on, and science itself is kind of redefining itself. And the idea of whether or not you can make biology predictable is very tantalizing, but it seems to constantly end up with people's noses getting smashed against the glass at this point, yeah. I would say.
And some people recognizing that. I love the passage where you're talking about this sort of this brainstorming session where it was essentially, you referred to it as jazz, essentially, what these they were trying to sort of understand in the early days of understanding genomes, right? It was so...
Metagenomes, yeah.
Metagenomes.
They were just, it was like 6 people in a very airless room looking through 3 or 4 databases at literally millions of genes. And they would switch from one database to another, and the people sort of started to meld. Like there were 2 very high-level people, one of whom had a photographic memory. And so she could look at protein codes and she could tell you what their function was, which is nuts. I mean, like, that's not normal. And then there were 2 postdocs sort of running the databases, and then the
projecting stuff all over the room.
Projecting stuff all around the room, and then they were kind of making up stories within the termite and trying to figure out what they meant. And 40% of the genes had no predicted function at all. So they were without, it's like a logic game without 40% of the information, and then the 60% that you have, it's unknown how much of it is totally false. In fact, we'd frequently get to the end of a tree, and the lead researcher would look up and say, "That's ridiculous. Go back," and then we'd go all the way back.
Yeah.
So yeah,
that's a great scene. Okay, questions.
So you raised some really profound questions. So what was your answer to what does it mean to be human?
Well, I think one of the things it means to be human is that we can solve certain kinds of problems with technology, but we don't necessarily solve the human problems of justice or what is right. And so one of the things that we need to do, first of all, when you make technology that's supposed to solve a problem, who decides what the problem is, and who decides how the technology is going to solve that? That tends to actually get done by scientists, but then to get the thing out into the marketplace or out into the world, there's another world of whether or not the technology survives in the market. Artemisinin, the malaria drug, did not, even though it had a lot of funding from Bill Gates. And if that can't survive, it's hard to imagine how other things are going to make it out to the world. So that's one thing. But the other thing is, we're constructing a future for ourselves, much the way the termites make a mound, but we don't have any rules for it. And we're not looking at the global thing that we're constructing at this point. And individuals do not have any say. And so we actually need to sort of start talking about the future that we want, and then trying to make the technology, thinking about how you get technology that gives us the future you want, because otherwise you're just going to get a future that you might not want at all.
But I also think the lesson is a little more depressing that I took from the book, which is that actually you can't really understand what you're building. You could sit down and say, we should all sit down and have this rational conversation, but in fact, that was one of the lessons from the book. Is like those tend to be those tend to be wrong. They tend to be
well, or what you think you want in 1971 is going to be different than what you want in 2011 and 2051.
Yeah, but even if you're, I mean, you touch on climate change throughout the whole thing, and even if you know that you want to, limit climate change, it's not obvious whether the actions you're taking are going to be helping you or not, just because there's so much complexity.
Right.
It's so easy to misunderstand what's like. Getting back to understanding
the termites themselves play a role in climate change, and they play a role in mitigating it. And nobody's really studying the termites for that.
Right. Yeah. And understanding the architecture, just why and how they build their mounds. It's like, what are they doing? Is this about those we recognize that these ideas come up. We think we understand them. We're starting to build a larger picture, a larger understanding, and then all of a sudden it's like, oh wait, we didn't. That sort of we realize that was a house of cards. That particular idea.
Yeah.
So more questions.
Thanks, Dan. Yep. Hi, my name's Andrew Dufresne. I'm a science educator and communicator. So playing off some of these questions, since we're staying with the high order ones, as someone looking from the outside in, but also seeing the technology and science develop firsthand, and then seeing the scientists come to terms with that, the progress or failure of a project, do you think our increasing understanding of the minute and the tiny and the previously ignored does get us closer to seeing that commonality in our humanity, and that perhaps we're actually, even as humans, not that different than a lot of the other things that are alien on our own world? Do you see that there's hope that we'll actually, through this investigation of the science behind everything, come together? And do you think writing something like this helps that? Or do you have reason to
well, I
that?
certainly had hope that, yes, that people, that in showing the science in a slightly different way, I could create a way of looking at it that encouraged us to see it in a human way. But I also have to say that I think that we're like woefully uneducated in science. And one of the things, as I was sitting in that meeting where the geneticists are cruising through the databases, I realized, no one should get out of high school. Now we learn a little Shakespeare, a little economics. You shouldn't be getting out of there without understanding some genomics and how to read a phylogenetic tree, how to read, there needs to be, we need to understand more of these complex sciences. They can't just be locked up in the labs. We actually need to understand how they work.
Just on that, to what end? Because I agree with you that the science knowledge should have a broader base. That we need to have a more scientifically literate public. But I want to hear your opinion on as to why, given your experiences.
Okay. Well, I think that right now there is a very divided discussion of technology, with some people saying this technology is okay and other people saying that technology is not okay. We don't have very deep discussions on it, and we deal with it almost entirely through concepts of utopia, like this will solve our problems, or an incredible dystopia apocalypse, like this is the apocalypse. And if you're a science writer, much of what you frame for even very good magazines, they will ask you to say, for example, I did an article on methane hydrates, very complex. They wanted to know, is it going to fry the planet or save it? That modeling of technology it makes it impossible to discuss the details of this is, no, but it's going to do this, this, and this. Or the power systems enabled by this particular technology may not be what you want. How would you, react to that?
Journals ask us to do the same thing. Discussions. It's got to cure cancer, or
yeah, right. It's a very common framing of things, and it just brings us back to stories from Frankenstein, basically.
Hi, Darcy Gentleman. I'm a presentations coach and science communications consultant. I wonder if you speak to how the concept of parasites has changed from a nuisance that needs to be dispensed with to something that's very good at surviving and inspiring technology or genomics research.
I mean, the simplest example is the fecal transplant, which if I had told you 15 years ago people are going to be asking their neighbors for fecal material, which they then put inside themselves, you would, using a blender, you would say, wow, you got problems, you need to leave. But that actually has happened, and that's been through the study of the metagenome of our guts, and through a limited number of clinical setting uses of that fecal transplant. This has become a cultural phenomenon. There's, I listened to some podcast about some woman who it combined like internet dating with fecal transplant. It was unbelievable. And that's sort of how our culture is processing this stuff. So what used to be thought of as a parasite, the things in your gut used to be thought of as parasites are now seen as some sort of a savior in a way, and you have people eating in ways to change their gut metagenome. You also have researchers doing very questionable science in, say, there was a guy in Africa who was studying people who he thought had a more sort of essentialist paleo diet, and he wanted to transplant some of their fecal material into himself so that he could then grow it himself. Which is also like, ideas from the 1700s that suddenly come through genomics and shoot out the other side into total weirdness. So, I think that the whole concept of the parasite has changed. Yeah. I'm sorry. I shouldn't be calling. You should be.
I wanted to let you finish it too. I'm sorry. Jumping off from there, my name's Jim McCormick, by the way, and I'm trying to study human cognition.
Okay.
This is really cool. And I don't have a word for this, but there seems to be a space where the way you act in ignorance is one way, and then as science and you learn how things work, it looks very different. And then when you really know deeply and intuitively how things work, it starts to look kind of like it looked like when it was ignorance. Mm-hmm. And there's a bridge you have to get over to teach the ignorant how to do it by the discipline and the rigor, and then how to let go of it and loosen it up and do what really works. Did you find any of those examples in the termites or in these structures?
Yeah, there's a whole interesting role of intuition. Like, what is human intuition? How does the human brain work? It's kind of a termite mound itself. It's somehow the structure of the brain limits and enables what we see. So, one of the most interesting things was there was an ecologist and a mathematical biologist who were out in a field together. And they were trying to figure out the influence of termite mounds on soil fertility, because the ecologist had measured the geckos. And when you measure the geckos, you find that there's a lot of geckos on top of the termite mounds and not so many further away. And that means that the geckos are somehow, the termites concentrate or build these sort of islands of fertility in a polka dot pattern all over huge fields. Maybe all over the world. They are remodeling from underneath in the very close to the equator. So he saw just a polka dot pattern. The mathematical biologist looked out at the same field, and she felt like she saw another pattern at the same time. And what she saw was leopard spots, which actually are like a Turing pattern. And so there's a way of sort of predicting the occurrence of leopard spots, and that would indicate that there were different functions working on the grass. So the ecologist he was like, no, you don't see leopard spots, that's just root competition. And because he knew too much. And the mathematician said, well, why don't you pull the Land Rover over and I'll stand on top of it and look out at this burned field. And she was like, I think I see leopard spots. So they ended up doing a lot of analysis, and she did see polka dots plus leopard spots, which meant that there were two systems, there were two interacting systems that they could model, and that could show how this fertility was working. It was very interesting, and then it turned out to be, they could extrapolate it to aerial photographs, and so yes, intuition at a high level is useful, but at the same time, it's also very misleading for scientists. So they're constantly balancing that. The roboticists, one of them muttered under her breath, there's absolutely no such thing as intuition. Yeah, exactly.
Thank you. Interesting topic. One of the central themes seems to be chaos theory or complexity theory in all of the different realms that people have talked about. And so I'm wondering, do you see science or education looking into chaos theory itself as a way to understand all of these phenomena, like the leopard spots example? It seems like there's mathematical entities going on there. So my question is, are you saying scientists and educators take that on as a topic in and of itself, regardless of whether it's termites or whatever?
Yeah, I think complexity theory is really interesting, and it's being used all over in labs in different ways. Not so much chaos theory, but the complexity theories. It's very difficult, though. They call it that they're waiting for Carnot, which gives you some idea that it's the play waiting for Godot. They're waiting for someone to show up. They're waiting for these big insights to show up, and they would be like Carnot's theories of thermodynamics. And so we're not there yet. Complexity theory is well worth studying and kind of fun and heady. But people who've went into it 10 years ago have come out the other side saying, oh, we're not going to understand the brain in my lifetime, and so I need to move on. And that's how some of them got into robotics and other things like that.
Yeah. So do you think Carnot will ever show up, or is that a misplaced aspiration?
I kind of go both ways on it. Sometimes I think, yes, there might be some insights that connect. The big problem is that, are you going to look at the field from the level of the giraffe? Or are you going to look at the field from underground with the termites? The part of the problem with complexity is that it's really about where you decide to situate yourself, and the complexity just spreads out forever, and it goes up and up and up, and it goes down and down and down. And so there's, I think
I had that sensation reading your book. I remembered this movie and book actually that when I was a kid in elementary school, they played this Powers of 10 movie where you sort of you start at our normal scale, and you go down and down at the atom and out to the universe. And I sort of thought, where should I be standing to fully appreciate this?
Exactly, even where in time. Right. Should I be a tortoise or should I be an E. coli?
Yeah. And the answer is you have to stand everywhere, or you're not going to understand it. But that was sort of fun to have that image.
And actually, one of the scientists in the book spends time going through those powers of 10. There's something on the web. He was like cruising in and out of it while talking to me.
That's a great thing. We think about it.
Hi, Ben Schaus from the Pew Charitable Trusts. Lisa, you mentioned we don't want to go back to the old Frankenstein story or the old formulas, but I've also heard the theory that there is only one kind of story. It sort of follows rising action and then it resolves. Everyone sort of wants this pattern when they hear a story. But how do you broaden that out for when those editors come to you and say, no, it has to be this or this? And more importantly, readers, they crave that. How do you broaden out and find interesting stories that aren't in that predictable mold?
Okay, well, I think this is really after my heart, but I think that you can take a story where you take someone's preconception and you take them in the little arc and you say, okay, well, this is where we are, and then you tweak it and you say, but look, it's like this from the other side, and then you build another arc and then look like this. So some of the reviews of this book have said that it has a terrible structure. But actually, I think that it is absolutely essential that you keep looking at your preconceptions and moving on. I mean, it's a series of arcs until you get all the way around the thing, and then you have a more complex sense of what you're looking at. So, yeah, I mean, try to go on a tour of the elephant's foot, I would say. Yes.
Kevin Finneran with Issues in Science and Technology. At the beginning, you talked about when they first started looking at the termites, they found that it was, that there were types of organisms that they weren't aware of that were playing an active role, and that somehow these genomes were also interacting or working together in a way that they hadn't anticipated. Was that really new science, or just that they didn't know this was happening in termites. So in other words, was
Oh.
this, in looking at a practical problem, did they actually come to a very important fundamental scientific understanding or new knowledge? And
Okay, so
and then if you want to speculate from that, did this make you think at all about whether or not, in trying to advance basic science, sometimes taking an applied approach to problem solving might actually be the best approach or best avenue to making fundamental discoveries?
okay, I can talk about that from two different. I can't make those questions seek together, but I can give you the first one. So actually, Mixotrix of Paradoxa, that one of the protists, became famous because different microbiologists thought that it proved a theory that originally legs on microbes might have actually been separate organisms and become incorporated. So it was similar to what happened with the mitochondria, which came into the cell as a free-living bacteria, came in and settled in and became integrated. And then gradually they've done research on it that shows that it's not. But there are a lot of papers that take that speculation. And so a lot of people followed that idea because it was a very attractive idea. This idea that that we had, that maybe my leg was originally a shark, and that it was different creatures that came in and attached and began to collaborate, and that's how we became complex. But that's only part of the story. And so there was the story built up, and then people began to dismantle it. So that's more the sort of what's been discovered, is that things are very freakish in the termite gut, and freakishness is enabled that isn't enabled anywhere else. I think there's another thing that's starting to evolve. There's an understanding of metagenomes as the level that you should be looking at, like gut contents, that they're no longer the individual organisms that they appear to be. Spirochetes have lost their sniffers and they've lost their motility genes. So how is that a spirochete? It's not a spirochete, but it still looks like a spirochete and it still has some spirochete markers. So they're having to kind of rethink what this heavy-duty symbiosis means in terms of the way we define individual species of microbes. So that's actually a huge insight, and we don't know. I mean, it's barely an insight right now. It's like the sort of thing that people show on their PowerPoint slide. Lead PIs show it on their PowerPoint slides, and then in 5 years maybe we'll see a paper on what that, more of what that means. So that's one thing. And then your second question was about applied science versus.
Right. Well, you looked at people coming from very different perspectives and looking at a problem. And some people, including Dan, have said that maybe science would advance more quickly if we looked at things in a more applied way. If we had, that Pasteur's quadrant type of work was more prevalent. But in looking at these, people who saw things very differently, how much value did you see in taking an applied perspective to what turned out to be a basic scientific problem?
I think the applied perspective was pretty successful in here. I mean, it didn't result in the technology or the solutions anyone was looking for, but they certainly had a lot of insights. But I think that there's a benefit also to the non-applied, the people who come up with weird, unwieldy ideas. Those send people into more interesting questions, I think, than kind of monkeying around with small questions around the edges.
I actually thought also the collaboration was there was a synergy there that made a big difference too. I think it wasn't just like who's right, but I think they were trying to play together.
Yeah.
And I think that provided insights. Yeah.
Can I ask another?
Sure.
So, of both of you, I think, because sort of science communication has been a theme in some of these questions, and you kind of gave a fairly standard answer, at least, about how only kids in high school learn more about genomics. But then it seems like you're saying something really different, right? So science communication has become a bit of an obsession. The academy's obsessed with it. There's all sorts of committees going, it's a big enterprise. You guys are my favorite science communicators. It doesn't seem like this question of the complexity of science and the complexity of our future is being communicated in a way that helps people get at it. So are there things that should be done differently, could be done differently, where to intervene?
Yeah.
You first.
Yeah, you first, Richard.
I want to save some for you. Yeah, I think we are kind of, I mean, human beings are by nature storytellers, and I think we can't get away from framing things as stories, and we need to. But it is also true that that's not the way the world works. I'm thinking about these drawings of these incredible metabolic pathways, right? And you can't tell a story about the Krebs cycle, right? It's like, well, you can, but it's not a linear story. It's, and I think that's, and a lot of what you outline is how much it's hard when you start to take things apart and look at them in individual pieces that they make less sense, and you have to realize how much everything is integrated together. And it's not, the answer is not as the termites or the gut bacteria or the fungi or even the landscape that may be helping to shape the termite mound. But so we're kind of stuck with the narrative structure one way or the other. And the question is, how do you take narratives and tell these much more complicated stories? I think this book does a good job, but I also think that realistically, it's hard to boil this down into a single bite-sized article, right? That's the problem. You need someone to take the time to sit down and take an afternoon or a couple of afternoons to say, okay, what is, to lean back and understand this complexity. I think so. We're kind of, we as a modern technological society are moving in the other direction. If you can't say it in a tweet, it's like it doesn't matter anymore. So I think that it's really, in some ways, it's a question of attention span and recognizing that we have to work somehow with stories. But yeah, I think I don't think you can do some of that in a classroom for sure if you get kids to think about this stuff earlier on. But I would encourage that to be a much broader idea, not just simply you have to understand metagenomics, but you have to but getting to these ideas of complexity in interrelationships and understanding that knowledge is provisional, and so what we know now is not going to be necessarily what we'll know in 10 years, and we shouldn't expect that. So anyway, that's a rambling answer. Did it give you enough time to come up with a better one?
Well, I guess one thing would be to really I guess there's two things. One thing is, so could you do something, TED Talks are 17 minutes. People like that length for an idea. Like, could you get them in a room for 6 hours and have different people presenting different levels of complexity in some way to process that? I mean, could we do this together in some way that helps us learn and helps us start to think in terms of complexity? I think avoiding the narratives of apocalypse is a good start, and getting, one thing is actually getting science journalists to think a little bit differently, or science editors to think a little bit differently about how they present things. Or even presenting things as a problem rather than problem solved is another thing that we could do to kind of open up the narrative of how, the New York Times presents these terrible diagnoses of, someone was itching for 6 months and nothing would stop it, and it's like, but it ends, and everybody gets to jump in and talk about how to solve it. And we don't really, science journalism isn't really like that. We forecast we talk about somebody's working in a lab on a mouse, and it's all going to work out. And that's not, rather than presenting it as a question, I think that's one thing that we could do.
Yeah. And I think one way to do that is actually find interesting characters who are chewing at these problems, as you did throughout your book. It's like, let's focus on these people who are, are bugged by something and are chewing away at it. They may not get to their solution, but just watching them try, right? I think I think it's inspiring. Yeah.
I think also we need to rethink of who the scientists themselves need to rethink who they are in society, and we we have a tendency to view them as sort of like gods who run by a special system. And that may not be, you know, they've got laundry, they've got, you know, and they make mistakes, and there needs to be sort of more back and forth about how we talk about what they're doing and how they talk about it, without attacking them. And I don't mean to attack scientists. I just mean to sort of change the sense that they can make all the decisions for us.
One last question.
Last question.
That'll be good.
Exactly. I hate it when that happens. You know, it's the pressure. My name is Alexander Kravitz. I'm basically a pedestrian. I feel like I'm not a scientist. I'm not a writer. And but I have a question. So you talked about the importance of like teaching science in high school. And I was thinking, since we're in Washington, D.C., like I was imagining, if I'm a congressman, for example, and I'm a lawyer or, you know, something different, I mean, and I don't really have an understanding of this kind of fast moving, what would you say to you know, the White House has had at least, you know, like a science advisor, right? I mean, how can you educate policymakers who are not going to go for a PhD or, you know, spend 6 hours educating themselves about termites, et cetera, about issues where they have, you know, incidents? Mm-hmm. That's the question, right? So from high school versus, and the second question is, are you thinking about your next book? I mean, I'm just curious.
Okay. That's actually a big question for Dan, is how do you talk to policymakers? I would say there's two things. One thing is, you know, scientists in the state where the policymaker is from should start to reach out to them and talk to them and create kind of a bank that they can go to with questions of people that they trust. That in California, that is very, very highly developed. UC Berkeley and the other UC system scientists are all really networked into the policymakers in the state, and that's why the state's policies move so fast on greenhouse gases. It's very dangerous for the policymakers, and it's very dangerous for the scientists because they can become politicized, and the UC system could suffer in many different ways, but that is part of what's going on in California, is that there's a very tightly linked network, and those networks actually start at the grad school level. I mean, I know someone who's now a state senator in California who formed a group of engineers and scientists to talk about climate change while he was a grad student there. And the network is, plus it's venture capitalists and the network is like 500 or 600 people, and they remain in touch. So if you want to do it the way California does it, I think. And then the second thing is, if I were a policymaker, I would be very focused on talking to people in my community about what they want for the future and looking specifically at what the military is funding for technology. That is one place where the weird thing about the military funding technology is that it's one place where democracy, or a representative democracy, could actually step in and make a difference on a technology, because you can't really do that in the market. But you can actually make a difference in terms of military technology. So that's one place that's... Oh, the second book, yeah. Yeah, so people keep presenting me with different ideas that they think are like more boring than the termite, but I could make them exciting. You know, clams has come up. My sister's like, clams, you should do clams. I'm like, Another friend said, you know, economists and theories. I was like, oh my God, I don't know. I said, I'm not sure. I'm barely recovering from this one.
Yeah.
We'll end on a note of uncertainty. So, Richard, thank you for your beautiful way, teasing out so much of the richness of Lisa's book. Lisa, thank you for your obsession, for sharing it with us, for the difficulty of turning it into this beautiful book. Thank you all for coming. Mahmoud, what's our next thing? Do we have something on the calendar yet?
End of October. Well, October 9th.
Oh, October 9th. We're going to have a big, very cool open house here October 9th with a public deliberation around autonomous vehicles. Ooh. And a release of our new book on new science policy tools for making the world a better place. So you'll be hearing about that. So thank you all for coming. Thanks, guys. And books in the back.
Thank
So
you very
see
much
all
for
next
coming.